This is Suganti, Assistant Professor, Department of Biotechnology, Vistas, Chennai. In today's class, we are going to see in detail about prokaryotic and eukaryotic translation mechanism. This is the structure of eukaryotic cell. In this, you can see the where the transcription and translation process will occur. Actually, in the nucleus, the transcription, the conversion of the DNA to mRNA will take place. Then after that, the post-transcription modification also takes place in the nucleus of the cell. Then after the processing of the mature mRNA, the mature mRNA will be transported into the cytoplasm. Here the tRNA will carry the amino acids and other ribosomes will help for the production of the proteins. So translation is nothing but it is the process in which the sequence of nucleotide triplets in the messenger RNA converted into a specific sequence of amino acid. During the synthesis of polypeptide chain. So translation is nothing but the nucleotide triplets in the mRNA converted into the specific sequence of amino acids. So in the last class, we have seen about the prokaryotic translation mechanism. What are the components required for the prokaryotic translation? Then about the charging of the tRNA before the initiation process. And also we have seen in detail about the ribosomes, the structure of ribosomes. And we have seen in detail about the initiation, elongation, and termination process. In today's class, we are going to see about the eukaryotic translation mechanism. So the eukaryotic translation is very similar to the prokaryotic translation, but there are few notable differences. So this is include, we all know that there is no signed Algerno sequence in the eukaryote. So instead, the ribosomal subunit recognizes and binds to the 5' capping of the eukaryotic mRNA. So in here you can tell like instead of the signed Algerno sequence, the 5' cap takes the place of the signed Algerno sequence. And also in prokaryotes, the formal methionine as the first amino acid in the every polypeptide. But here in eukaryotes, the ordinary methionine will be used. And also eukaryotes do not have a specific initiator tRNA. However, eukaryotic translation involves many more protein factors than the prokaryotic translation. In prokaryotic translation, we need only 2 to 3 translation factors for assembly of the ribosomes and other things. But here in eukaryotic translation, there are initiation itself, there are at least 10 eukaryotic translation factors are involved. So we will see what are the process. So the eukaryotic here like prokaryotes, here also there are 4 main phases of translation: activation of amino acid, initiation, elongation, and termination. But here the initiation process is little complex when compared to the prokaryotic translation. In prokaryotic cell, the transcription and translation are coupled. That means there is no separate location in prokaryotic cell. So both the transcription and translation takes place in the cytoplasm. But here in the eukaryotic cell, the translation occur in the cytoplasm of the cell. In a eukaryotic cell, the transcription occur in the nucleus, then translation occur in the cytoplasm. Then the steps are same like charging of the tRNA, then initiation, elongation, termination. But here the notable thing is the ribosome subunit is differ, and also the name of the helping factors for the initiation process also differ. And also here the 10 different eukaryotic initiation factors are involved in the initiation process. So first this is the first step of initiation process. So like prokaryotic ribosomal dissociation, here also the 80S ribosomes are dissociated into 40S and 60S subunit. So first the small subunit will be involved in the initiation of the translation process. two initiation factors named that EIF3. EIF3 means eukaryotic initiation factor 3. So EIF3 and EIF1A, these both initiation factors will bind to the party subunit of the ribosome, and thereby it will block the binding of the 60A subunit at the starting point of the time. But this 60S subunit, it plays a major role in the translation process. It will come and play in the later stages of the translation process. So once the 40S subunit and other initiation factors are coupled, it will form a 43S initiation complex. So it's automatically containing the methionine tRNA, then two initiation factors. Next step, automatically this complex is go and binds to the mRNA, and it will form the 48S initiation complex. So after binding to each thing, the name will be vary. The complex name will be vary. Initially the 43S complex, then 48S complex. Like here, the another initiation factors like 4G and 4A and 4E will come and play. Mm, Here the initiation factor 4F is called as a cap binding protein. It will bind to the 5' cap of the mRNA. Next, the ribosomal initiation complex scan the mRNA for the identification of the appropriate initiation codon. Wherever it find the AUG, it will start translating the amino acids. The last step in the initiation is the formation of the 80S initiation complex. Here, the 48S initiation complex binds to the 60S ribosomal subunit, the major ribosomal subunit. Here it will form the 80S initiation complex. For this binding, it involves the hydrolysis of the GTP and also the helping factor, the initiation factor 5. Next one is elongation. Once the ribosome elongate the polypeptide chain by a sequential addition of amino acid, the amino acid sequence is determined by the order of codon in the specific mRNA. So whatever the codons present in the mRNA, based on that, the tRNA will carry the amino acid and it will form the amino acid chain. Here, in elongation also some certain elongation factors will come and play an important role. Elongation may be divided into three major steps: binding of amino acid tRNA into A site, peptide bond formation, then translocation. And the elongation is similar to the prokaryotic elongation mechanism. Then binding of amino acid tRNA to A site. The ATS initiation complex contain methyl tRNA in the P site and A site is free now. The another amino acid tRNA is placed in the A site. So this requires the proper codon recognition on the mRNA and involvement of the elongation factor and supply of the GTP. So the elongation is same like first tRNA, it will carry the amino acid and it will sit to the acceptor side, then it will move to the P site, then side by side for the energy source, the GTP come and play, and another elongation factor EF1A will come and play. But in prokaryotic elongation process, there is no helping factors like EF1A. Automatically between the amino acid, the peptide bond will be formed with the help of the enzyme peptidyl transferase. So automatically the tRNA will bring one by one amino acid based on the codon in the mRNA. Then amino acid will be joined with the help of the peptide bond formation using the enzyme peptidyl transferase. Then the amino acid complex will be formed. Translocation is nothing but movement of the ribosome to the next codon of the mRNA towards the 3' end. So this process is called as a translocation. Translocation is nothing but the ribosome moves to the next codon in the mRNA. So automatically the site also will be vary from the A site to B site. So here also for translocation, some helping factors are involved. Elongation factor 2, EF2, and energy source also required to move the mRNA. Here the hydrolysis of GTP will take place. So here during the translocation, every second the around 6 amino acids are incorporated during the course of elongation of the translation in eukaryotes. Next one is termination. Of course, we know one of the stop codon or termination signal are occurred like when it finds UAA, UAG or UGA, the stop codon signal, automatically the growing polypeptide will be stopped or terminated. So the ribosome encounters a stop codon. There is no tRNA available to bind to the A site of the ribosome. Instead, a releasing factor binds to it and it will release all the tRNA ribosomes and growing polypeptide chain. So once after releasing, again the ribosome go to the next round of translation process. And also the mRNA, tRNA also released, and the ribosome must dissociate into small subunit and large subunit. And these events, the dislocation of the components of the translation is called as a ribosome recycling.